both molecular biologists and comparative biologists, seeking genotype-phenotype associations, and we hope that a lot of you will be inspired by the talks of this symposium. None of you want to listen to me waffle on, so I'll get right to introducing our first invited speaker, Nicola, who has a really exciting talk for you, I'm sure.
Okay, hello. Hopefully you can hear me. Thanks very much to the organisers for inviting me and for organising this symposium. So I'm going to talk a bit about when I think, sort of trying to find general rules about when we might be able to identify gene phenotype associations at the macroevolutionary level. I'm going to take a slightly autobiographical approach to this. So I'm going to start with work I did during my PhD, which was on evolution of sexually dimorphic colour in the galliform birds. So in particular, I was trying to answer the question of, could we detect rapid evolution of colour genes, so secondary sexual traits at the molecular level. So people had found that you could find there was rapid evolution of traits that controlled, or molecular genes that controlled reproductive traits, and this seemed to speed up when there were higher levels of sexual selection. But no one had really done this for secondary sexual traits. And the galliform birds were a nice group to work on because they have members that have really impressive male colour, like this peacock and these pheasants. And then they have members where the males are sort of brown and look quite similar to the females. So the idea was that in these lineages where the males look really bright, there's going to be stronger sexual selection acting on the plumage colour, and so we should hopefully see faster evolution of some pigmentation genes. So I looked at a bunch of pigmentation genes. One of these was the melanocortin-1 receptor, which some of you might have come across before. It controls colour differences in a range of vertebrates, sometimes to do with camouflage, but other times to do, it seems like the traits are maybe sexually selected. So I looked at this gene and some other pigmentation genes and a couple of controls. And what I found was that the MC1R gene was evolving faster on lineages where there were higher levels of sexual dichromatism, which I was using as a proxy for the strength of sexual selection. So that's what you can see here. This is like a reconstruction of the strength of sexual dichromatism. And on the branches where there were higher sexual dichromatism, there was faster evolution of MC1R using the ratio of the rate of non-synonymous to synonymous substitution as a proxy for the rate of molecular evolution at this gene. And even though this was what I'd really set out to test and what I'd hoped to find, it still kind of surprised me that I found this, because this suggests that not only is this gene evolving in response to sexual selection on one or two of these branches, it seems to be doing this repeatedly across the phylogeny. So this really sort of started me thinking about when and how often do we expect to find this pattern that the same genes are repeatedly targeted by natural selection, which is essentially what needs to happen in order to see this sort of thing occurring. And then if we really want to look at gene phenotype associations, we actually need not only the same genes to be repeatedly targeted by natural selection, but to be producing the same phenotypes. And in a way, that's not really what I was looking at in the galliforms. I was just looking, kind of using dichromatism as a proxy for the strength of sexual selection. But maybe, if we want to look at gene phenotype associations, we would really need this sort of repeated evolution of the same phenotypes. So how often are these criteria met, and do we expect genetic convergence to be the rule or the exception? And there have been quite a few people that have written reviews about this and tried to answer this question, including this quite nice meta-analysis. But I think essentially the conclusion was that we still don't really have a good idea of the answer to this. And in particular, some of the ways that we identify these genes that are evolving in response to natural selection could be sort of biasing us to repeatedly finding the same genes. So moving on from working on galliforms, I moved to the heliconius butterflies, and they seemed quite a nice system to address this question, because we see this really close convergence between distantly related species. So all of the species on the right here are Helicones melpomene, all of the ones on the left here are Helicones erato, and wherever they occur together, they converge on an almost identical wing colour pattern. And this is because these are warning colours, so this is Müllerian mimicry, and they're sharing the cost of teaching predators that they are unpalatable. But not only do we have this really close convergence, we also see this huge amount of diversity within species, which then allows us to cross them in things and identify the genetic basis of these traits. So I'm not the only person who's been working on this system. Over several decades, people have been working on identifying these genes, and the outcome of this has essentially been the identification of 5 major genes, so at least 4 of them are identified to the particular gene. This one's still a bit unsure. But these 5 genes that essentially repeatedly across the phylogeny control changes in colour pattern at the microevolutionary level. So each of these is one species, and the diversity of colour patterns that you see within that species. And then in some cases, like Helicones erato and melpomene, which is what I showed you on the previous slide, these 2 distantly related species tend to use the same genes to control similar phenotypes. So within each box are examples of the types of phenotypic change that gene produces. And you can see that in general, in those 2 species, the genes are producing similar phenotypic changes. Whereas in these other species, the same genes are involved, but they're producing sort of different phenotypes. So even though we have this strong level of convergence, we have those genes sometimes controlling the same phenotypes and sometimes controlling kind of different phenotypes. And in fact, this extends even beyond Heliconeus, so to the peppered moth, and this was work that was done by Aaron van Toff and Illik Zakeri in Liverpool. And I'm sure you all know about the peppered moth. And this is the cortex gene here. And what they found was that in the black peppered moths, there had been an insertion into one exon, an intron of cortex that had caused the peppered moths to go from being their white speckled colour to being black. And this, if you noticed on the last slide, this is one of the genes that also controls colour pattern in Heliconeus. So it controls colour patterns in erato and melpomene, as I said, and also in these other species that were on my last slide, and then in the peppered moth, and also in the silk moth. So even over these much longer evolutionary timescales, some of these genes are controlling colour pattern differences. So this is kind of repeated gene use at the really big macroevolutionary scale. But also within Heliconeus, there are some kind of possibly maybe unique things to the Heliconeus system, or maybe things that we're realizing are slightly more common. But in particular, these butterflies seem to hybridize with each other a lot. So this is aiding gene flow between the species and allowing them to share alleles at these color pattern loci and aiding convergence. So some work I did with Kanchondasmopatra, we showed that at the sort of background, so most of the genome, this is essentially the species tree. We have Helicones melpomene, Helicones timoretta, which mimics melpomene, and these are now closely related species, so much more closely related than erato and melpomene that I mentioned previously. And then we have this other group here, including this species Heliconeus elevatus, which looks a bit like melpomene and erato. But then if we make the same phylogeny, but at the locus that's controlling, or at one of these loci that's controlling color pattern, in this case, it's the gene optics, which was one of those 5 genes that I showed you at the start. This phylogeny switches around. So we have melpomene and timoretta that mimic each other at the top, grouping together. Again, these other 2 populations of melpomene and timoretta that mimic each other are grouping together. And we even have Helicones elevatus, which should be down here, grouping up with melpomene and timoretta. So this could either be due to shared ancestral polymorphism at these genes that's been shared across evolutionary time, or due to hybridization, and combined with a bunch of other data, we really think that these are hybridizing and sharing the color pattern genes. And this kind of gives us a tool to identify these genes. So actually using these patterns was one of the ways that I identified the cortex gene as controlling the yellow color pattern. So we could look for associations between sort of species and populations that shared the same color pattern elements. And this is now a sort of small region, a kind of 1 megabase region where we knew one of these genes was. And then look, these at the bottom, this shows associations between genotype and phenotype and the strength of those associations. And so if we look for associations with the yellow hindwing bar, which we knew was controlled by this locus, there were SNPs that were essentially perfectly associated with this color pattern within one of the introns of cortex. And then if we look for associations with this yellow four-wing bar, which we also knew was controlled by this locus, we find other associations upstream of this, likely controlling, corresponding to a regulatory region. So we have kind of this convergence resulting from hybridization and ancestral polymorphism of the same alleles. But we do also have the other situation of repeated or separate mutations resulting in convergent phenotypes from this gene. So if we now go back to looking at Melpomene and Melpomene and Erato, which were those distantly related species, so they diverged about 10 million years ago. These are the associations, that's the association with the yellow hindwing bar in Melpomene that I showed you. If we look at the same association with the yellow hindwing bar in Erato, they're in slightly different positions. And in fact, in the two populations of Erato that both have this bar, so one's in Panama and one's in Peru, and they both have this yellow bar, they seem to have evolved this phenotype independently in these two populations. So in the Panama population, we again have associations in an intron of cortex, but a different one. And if we look in Peru, they're quite far upstream of cortex, the strongest associations. So suggesting that these are two different alleles, and that even at this close evolutionary timescale in Erato, we've had independent evolution of cortex. So one thing about this is that obviously these alleles that we're looking at are fairly major effects, so they mostly control turning on and off of these major color pattern differences. So one thing that might be a possibility is that when we have these sort of major effect loci, maybe those are more likely to be reused than more minor effect loci, so things that control small changes. So we looked at this in, again, Erato and Melpomene, so these two distantly related co-mimics. And as well as having major differences in pattern, they also have some more minor effects. So here you can see they have this red bar changes from being quite small, this is the Ecuadorian population, to being quite wide in Panama. And we have crosses between these, and I had a master's student, Hannah Bainbridge, who looked at the variation of this bar shape in F2 crosses using the program Patronize. And so you can see the size and shape varies. And then she did a multivariate QTL analysis of this shape variation. And what she found, so from fitting different fitting models with different numbers of these loci, there were only two loci that came out as being significant in Helicones erato, one that was on chromosome 13 and one that was on chromosome 18. And in Helicones melpomene, there was just one on chromosome 18. And so the gene optics is present on chromosome 18, so that was the first of those 5 genes that I showed you. And then chromosome 13 is this locus that's been found before, but we don't necessarily know the gene. So even for these more minor effect things, we're still finding the same loci, and we're still finding the same loci when we compare convergent phenotypes, convergence in sort of quantitative phenotypes that we might expect to be controlled by more genes that might have less constraint on them. And then, so as well as looking at this multivariate shape analysis, she also looked at just the area of the band. And then, in fact, you get even a higher level of parallelism. So in Heliconius melpomene, we now also get chromosome 13 coming out. So essentially, in both of these species, we've got chromosome 13 and chromosome 18 controlling most of this variation. But maybe this isn't the whole story. So you might have noticed that in these crosses, we also have another trait segregating, which is this blue color. So it's quite obvious in Heliconius erato. It's slightly less obvious in melpomene, but it is still there. You may or may not be able to see it. Mel Bryan, PhD student in my group, has been analysing this blue colour variation in these crosses. And this is what it looks like in the F2s compared to the distribution of the parentals. And so you can see it's pretty continuous in the F2s, and there's very little overlap with the parental values, so suggesting that this really is a polygenic trait. It's controlled by multiple loci, and then if we do a rough estimate of the number of loci, it comes out as being about 7 in erato, and about 5 in erato and about 7 in melpomene. And this blue colour is kind of interesting, not just because it's this quantitative trait, but also because it's a structural colour. So unlike the red and black and yellow sort of patterns that we see, which are controlled by different pigments, this blue colour, if we zoom in, these are now the individual scales on the wing. These are individual scales. You can see these ones look blue, these ones look pretty black. If we zoom in still further, in the black one we can't see anything because there's so little light reflected. In the blue one you can see these blue lines that run down the scale that seem to be sort of dotted. And then if we look at it under SEM, we can see that in the blue scale, or in both scales, there are these ridges that run down the scale. In the blue scale, these ridges are very flat and overlapping, whereas in the black scale, they're curved and they don't really overlap. So this overlapping structure acts as a multi-layer reflector, and this is what produces the blue color, whereas in the black, essentially all the light passes into the scale and is absorbed by the melanin that's in the scale. So we're sort of interested in how this iridescent color evolves compared to the black color. And if we look across the phylogeny, this blue color seems to have evolved multiple times. And it's produced by, in all cases, it's produced by these ridges and seems to vary depending on the curvature of the ridges, but also the spacing of the ridges. So if these ridges are packed more tightly together, then we have more of the reflectors, which is what you can see here, and then the reflectance is higher. So we get this relationship here. And if we look at co-mimics, so erato and melpomene, for example, I was talking about, you can see they have very similar ridge spacing, so they seem to be converging on ridge spacing. This is another co-mimetic pair, so Sidno and Elucia, and again they have similar ridge spacing. But you can see they don't really have similar reflectance. So the best explanation for this is that they are packing the ridges together more, but the ridges are just not as good reflectors. And you can see that there actually seem to be two different slopes to this. So if we look within the erato clade, it looks like as they pack the ridges together, they get brighter faster. Whereas if we look in the melpomene clade down here, they don't get as bright as fast. So essentially in the melpomene clade, the ridges are not as good reflectors. They're more curved, they're not as overlapping. And in the erato clade, they seem more able to evolve these really flat, overlapping ridges. So overall, this seems like ridge spacing can evolve quite quickly and is not very constrained, whereas actually the ridge structure, the flatness and the curvature and making these ridges really good reflectors seems to be much more phylogenetically constrained and something that's a lot harder to evolve. So what will all this mean in terms of the genetics of the trait? And so Mel's also been doing this, QTL mapping of this trait. And And what she finds is essentially there are different loci that are controlling the colour difference in the two species. So in Melpomene, there is a sort of medium effect locus on chromosome 3, whereas in Erato, it's sex-linked, and the largest effect locus is on chromosome, the Z chromosome, and then there's also some effect of chromosome 20. And even if we look down at more minor effects, there really seems to be no correlation between the effect size of loci, of sort of homologous loci between the two species. So it really looks like there's very little sort of genetic parallelism in this kind of more complex trait, possibly because of this higher sort of phylogenetic constraint that we seem to have, and the fact that it seems to have evolved quite differently in the two species. So I think overall it suggests that sometimes we've got the same genes controlling the same traits, other times we don't, and really predicting this is quite hard without knowing quite a lot about the details of the trait and how it's produced and how likely it is to be controlled, to be subject to things like developmental and genetic constraints. So overall, I would like to thank the people who've been involved in all this work. There's obviously a lot of stuff I presented. I think I thanked the main people during the talk, but just to say that Mel, who did that QTL analysis of the iridescent colour, just handed in last week and is now looking for a postdoc. Thanks very much.
So thank you for a good talk. I don't know if this is on, but has anyone got any questions?
Great talk. I was just wondering what genes were associated with those last QTLs that you were talking about.
They're quite big regions, so we don't really know yet. We do have some, so we've got some gene expression data, so we're looking at differential gene expression between the blue and the black butterflies, and we're hoping that'll help us identify some candidate genes. But yeah, we don't, they're too big at the moment to really say anything about genes.
What's the role of chromosome inversions in all of this, if there is any?
Apparently none, at least in the ones I presented. So the only exception to that, and I would have to go right back to the beginning, is Heliconius pneumata. So yeah, Erato, Melpomene, Timoretta that I was talking about. I went too far on this one. So basically, yeah, all of these, there seem to be almost no inversions. Heliconius pneumata, where we have one locus that essentially controls all of the variation, there's a big inversion. So Paul, I don't know if he's in the audience, he spoke day before yesterday, and he thinks there are multiple genes within this inversion that are controlling it. But yeah, I don't know. I think maybe the jury's still out on whether it really is just cortex, like I've shown here, or whether there's multiple genes. But yeah, he thinks maybe there's multiple genes.
So thanks for the talk. One of the QTL was on the Z chromosome. Could be that this trait is also linked to sexual selection in that species and not in the other?
Yes. Yes, it could be. So Mel had a poster in the first poster session. She's also been looking at sexual selection. So there does seem to be some sexual dimorphism. So males in Erato are slightly brighter than females, and that doesn't seem to be the case in Melpomene. So that might explain partly why Erato is maybe brighter. It could be partly due to sexual selection. She's also shown that, so she's done some sort of experiments where she stresses out the pupae, and the colour also seems to be condition dependent. But we're sort of in the process of doing mate choice experiments at the moment to see if it is sexually selected.
Okay, well, great. I'll just thank Nicola one more time.
heterogeneous traits like generation time or body mass, but I will concentrate today on discrete traits, meaning if you categorize a species to habitats, so we can look for saltwater habitat versus freshwater habitats. We can look at pathogenicity, bacteria that are pathogen or not. We can look at reproductive modes of plants versus sexuals versus asexuals, crossers versus selfers, etc. And for simplicity in this talk, I will concentrate on binary traits, meaning traits that we can categorize to 0 or 1. But obviously we can extend such methods to categorical traits, 0, 1, 2, 3, etc. Okay, and the basic questions that we are interested to ask is, do repeated transitions in organismal traits lead to alterations in the patterns of genome evolution? So just let's look at quite a famous examples by Smith and Donahue that looked for rate variation or substitution rate variations between trees and herbs, or basically a woody plant versus a herbaceous plant. And in this study, they analyzed several large clades of plants. So here we can see 2 examples. And they mapped the brown branches. Those brown color branches in the tree are woody species, and the green branches are herbaceous. And what you can instantly see from this figure is that all the green branches are much, much longer, right? Meaning that trees or woody plants evolve much slower. The woody are shorter, so the woody plants evolve much slower than herbs. And in this study, the authors claim that the generation time of woody plants that are longer leads to the slower rate of evolution. But obviously, there are other reasons beside the generation time that cause woody plants to evolve slower than herbs. Okay. But when we see such rate variation owing to a categorical trait, it can also change our predictions in several ways. First of all, we know that the long branch attraction phenomenon so it can cause the phylogeny to group slowly evolving groups together and fast evolving lineages together. Similarly, we can get errors in divergence times estimations, and just as pointed by Smith and Donahue, relaxed clock methods are likely to estimate that slowly evolving groups are younger and that rapidly evolving groups are older than their true ages. So how can we would like to integrate species categorical traits or species characteristics into phylogenetic comparative methods? And we would like to do it in several levels. First of all, we would like to reconstruct the phylogenies by taking into account also the phenotypes of the organism that we study. We would like to detect traits that repeatedly alter the substitution rate, and obviously to detect the genes that are most strongly affected. We would like to test for trait-associated alterations in the selective patterns. So most of us do KKS analysis, etc. And we can also maybe detect expansion and contractions of gene families that are associated with a certain trait that we would like to study. And this, most of the work I will talk about today was done by Eli Levy-Karin and Karen Halby from my group. Okay, and first I would like to concentrate on a method that aims to detect traits or regions that repeatedly alter the substitution rate of the organism that we study. Okay. So just as I mentioned, the study by Smith and Donahue that looked for substitution rate differences between herbs and trees. So these authors used the sister clade contrast to detect such differences. So what does this non-parametric sister clade approach does? First of all, it detects sister group contrast, meaning we would like to detect internal nodes in the tree in which one part is entirely green in this area, in this example, and the other part of the sister clade is entirely blue. And then we would like to do, for each contrast, calculate the average branch length from the tip to the common ancestor in here. And then we just perform a sign test across all contrasts. So we just assemble a large database, look for all internal nodes in the tree where we can find such contrasts, and we just said in how many contrasts the blue lineages win, and in how many internal nodes the the green branches win. Win meaning it has a longer or shorter branch length. Okay, but what is the problem in such a non-parametric approach? First of all, it ignores lineages not part in the contrast. For example, we only concentrate on this part of the phylogeny, but we ignore all this long branch. Second, we ignore the number of taxa within each contrast and the number of taxa between the contrasts. And another problem is that we assume that trait transitions occur only at internal nodes of the tree. But obviously, the transitions itself have occurred anywhere along the branch. So this method ignores a lot of uncertainty, and basically we know that sister clade contrasts have lower power. Another common approach, just as we saw in the previous talk, is to use a semi-probabilistic approach. What does it mean, semi-probabilistic approach? Let's say we would like to compare the substitution rate between the black branches and the blue branches here. Then before we do the comparison, we a priori specify the partitions, meaning we a priori said that that we would like to compare this whole branch against the rest, and the blue branches across this one, but we have to specify the partitions in advance. So we basically ignore also uncertainty in the evolution of the character trait. But once we make the partition, we use a likelihood approach. So we can assume that, for example, we compare the substitution rate, assuming that the whole tree evolves under a single rate, versus a model that we have one substitution rate for the blue branches and another substitution rate for the black branches, and then we do a likelihood ratio test comparison to say whether the more complex model is statistically justified. Okay, but this is obviously a mixed approach, so you use like a maximum parsimony in addition to a maximum likelihood approach. This is not the best approach we can think about. And as I said before, the categorization is done prior to analysis, and it is again assumed that the trait transitions occurs only at internal nodes of the tree, which is obviously unrealistic. Okay, so just to sum up what existing or previous approaches lack, first of all, we ignore, as of every maximum parsimony approach, we ignore multiple substitutions occurring across the phylogeny. We ignore back transitions. So to sum up, the number of trait transitions is obviously underestimated. And as I said, transitions are assumed to occur only at internal nodes of the tree. Okay, so the aim of the framework that I will present today is to unite the evolution of character traits with the evolution of the sequences or the molecular data that we analyze. So we assume that we are given a species phylogeny, okay, and we also are given the trait data at the tips of the tree. So let's say we have trait data is blue or green, and obviously we are given the multiple sequence alignment or the sequence data at the tips data. And our question in this method is to ask whether the sequence evolution rate in state 1 or in the blue state is different compared to state 0. Okay, so what are the model assumptions? First, we assume that the character evolution or the trait data evolves along the species phylogeny. Then we assume that the rate of evolution depends on the character trait at each point in time. And for both, so now we have two Markov processes, one for the evolution of the character data, one for the evolution of the sequence data. And we use continuous time Markov processes to model the evolution of both types of data. And when I said continuous time Markov process, we have a transition matrix. I will not get into the details, but for the character trait, let's say we have two states or binary trait, we have a standard two-rate matrix, two-state matrix. And for the sequence model, in case we are investigating nucleotide data, we can use only time reversible substitution process, Jukes Cantor, HKY, GTR, etc. Now, in order to unite these types of processes, we also add what we call an additional parameter, which we call R, or the relative rate, which signifies the rate of evolution of character state 1 relative to character state 0. Okay, so these are the model assumptions. Now, the problem is, how do we calculate the likelihood, the joint likelihood of this model, right? We have to somehow calculate the joint likelihood. And the problem is that when we use standard likelihood models, the likelihood of the sequence data is calculated for each position independently. But when we assume that the trait evolution affects the rate of the sequence data, then we must somehow take into account that the whole sequence is affected by the character trait. So we somehow have dependencies among sequence sites. So, and this is not a standard way to calculate the likelihood. But let's say that we know where the character transitions have occurred. And in this case, let's also know that R, the relative rate parameter, is 2. So we know where the character transition occurred, and we know exactly what is the relative rate ratio between the two character states. Then, so let's assume that we know that exactly here was the transitions and here was the transitions. Then we can multiply the blue branches by 2, which is the relative rate between the two states. So if we multiply the blue branches by 2, we got this new tree. And along this tree, we can calculate the likelihood of the sequence data, again using the standard methods where each positions evolve independently. Now, obviously, we don't know where the character transitions occur and what is the relative rate parameter, but we'd like to estimate this. So instead of assuming a fixed topology like, let's say, most branch state models, we use a sampling approach to sample many possible trees, or basically many possible character histories. For each character history, we do this multiplication, and we calculate the sequence data based on each one independently, and we sum up over the whole sample. The question is, how can we sample possible character histories? Obviously, we would like to sample histories based on their probability. And so I won't get into the details, but we use a method called stochastic mapping developed by Nielsen to sample the histories of character transitions according to their probabilities. And we do this using the model of character evolution. Okay, so we sample histories according to their probability. So just let's into the calculations a bit more formally. So this is 2 slides equations. So we would like to calculate the joint likelihood of the character data and the sequence data, given a model of character evolution, given a model of sequence evolution, the phylogeny, T, and the relative rate parameter, R. Using the chain rule, what we do is we calculate the likelihood of the character data, giving the character model, but we can drop several terms that we don't need. And this is just the regular Felsenstein algorithm that we calculate the likelihood of the character data giving the character model. But we multiply this by the probability of the sequence data giving the character model and the character model and the sequence model and the character data. So to do this is exactly the stochastic mapping approach that I just shortly told about before. So what we do is we do the likelihood of the sequence data is calculated by taking into account many possible histories. Okay, basically, we would like to take all possible histories of the character data. We cannot take into account all possible histories, so we replace the integral by sum, which is a stochastic mapping approach that sample histories according to their probabilities. Okay. So we call this model the trait rate model, which in this model we assume that rates of sequence evolution depend on the character state. Now we can compare this model to a null model in which the analyzed trait does not influence the rate of sequence evolution. Basically, we calculate the likelihood, this likelihood given the relative rate parameter is R. So this is our null model. This is our alternative model. And then we use a likelihood ratio test to compare the fit of the models to the data. And to decide whether the alternative model is justified. Okay, so let's look at a short example. In here, we looked at crustacean species living in saline environments, in saline water, or in fresh water. So in here, I color the species living in fresh water in red and in salt water in blue. And in here, we look at the non-coding mitochondrial sequences to look whether the genome-wide substitution rate of the saline sequences of the species living in saline environments is different compared to those of species living in fresh water. And what we got here is that species living in salt water evolves 1.5 faster compared to those living in fresh water. In this case, from the literature, we found out that species living in salt water, the DNA repair mechanism is altered by the high salt concentration, such that the DNA repair mechanism does not work properly. Okay, but in this example, we look whether the basically we looked at non-coding sequences, which indicates of the genome-wide substitution rate. But in some cases, we like to do an analysis gene by gene on a gene-by-gene basis, right? Looking for which genes are most strongly affected by the trait that we analyze. And in addition, in this method, we assume that the whole gene under study is affected by the trait that we analyze. But obviously, we know that if we look at a specific gene, some positions are maybe more strongly affected by the trait, and some positions are less so affected. Usually, those within the core of the protein are not really affected by the trait because they only usually the function is to maintain the structure. Okay, so next we extended this model also to allow only specific sequence size to be affected by the trait that we study. So again, basically what we do a mixture model where we assume that in each position we calculate the likelihood of over two scenarios. In the one we assume that the trait that in this position the position is affected by the trait, multiplied by the probability that the sequence position is not affected by the trait. And then we can do a site-to-site inferences and to infer which positions are most strongly affected, or basically most strongly affected by the trait, or we calculate the posterior probability of each site to evolve under the trait-dependent model. So again, just for an example, we studied orchids that some of them are photoautotrophs or able to do photosynthesis, and in some other cases, the plants are parasites or depends on other plants for photosynthesis. So in this case, we study a specific protein, the ribosomal protein S8. What was important here that this protein is retained in the parasites. So we can examine the sequence of this gene across the whole phylogeny, whether it's in the parasites or it's in the autotrophs. Okay, and in addition, a 3D protein of the structure is available, so we can map the site-specific inferences onto the three-dimensional structure of the protein. Okay, so these are the results. We colored, so this is the three-dimensional structure. we color the results according to the probability of each site to evolve under the trait-dependent model. So as the color is darker, the position is more trait-dependent. And what we see here is that, what we saw is that on average, the entire protein is affected by the trait that we study. And this is expected because the transition to parasitism increased the substitution rate of the whole protein. But what was interesting that the positions that are probably connected to protein-protein interactions with other proteins are those that seem to have the highest shift in the substitution rate compared to the rest of the proteins. Okay. So just to sum up this method, we have this method available as a web server where users can just download their sequence data, their trait data, and the species phylogeny. And then we do all the calculations. We project, if we have a three-dimensional structure, we project the rates on the three-dimensional structure or on the linear structure. And so I hope it's quite easy to use. Okay, so just to conclude, what I've showed so far is a probabilistic model that looks together at the evolution of the sequence data or the molecular data and the evolution of the phenotype that we would like to study. But obviously, there are other directions that we can continue from here. First of all, we currently would like to integrate this joint model into RevBase, and so to allow users to infer species phylogenies by taking into account not only the sequence data, but also possible organismal traits that may affect their evolution. And we extend this model to codon sequences. Karen, in this afternoon, will talk about how we can detect relaxation of selection or intensification of selection within such a joint model. And we also would like to extend this model to gene family dynamics to show whether specific gene families repeatedly contract or expand upon character transitions along the phylogeny. Okay, so I will end here by thanking my collaborators, the lab members, and the funding.
Thank you, Itay. Does anyone have any questions? Yep.
Hey, thanks. Your plans for incorporating the gene family evolution into the trait or joint trait evolution, what are you going to do? Like, what is your approach? Don't reveal anything, but just to have an idea.
No, we have some current methods, like if you know Café, for example.
Yeah.
That has basically, instead of sequence alignment,
I know, but
instead of sequence alignments, we have each position is the number of genes in the family, and then we can assume that and then you have the parameters of gene loss or gene gain. So we can change the parameters of gene loss and gene gain based on the character evolution at each point in time. So we can have either a single model where the rates of gene loss or gene loss and gain are just there is one rate for the entire phylogeny, or a more complicated model where the rates of gene loss and gains are dependent on the phenotype of the lineage. Okay.
Yeah, thanks.
Our extra plans, by the way, is to include also polyploidy into these equations. So in plants, it will make some sense.
Thank you. Nice talk. So how does this model handle unknown states or incomplete taxa, which is a common thing to come across?
Yeah. So for incomplete taxa, actually we can incorporate this, but just so it can. If you incorporate unknown taxa, it's basically like a gap character when you do like so. But it can incorporate unknown phenotypes. But obviously, I will not give a great support for this because it's not like in sequence data you have much data. But in here, the character data is really important. Mm-hmm, yeah. So in the analysis that I did, I filter out those species.
Okay.
Sorry.
So I have a question. How divergent can these sequences be? Are we talking about the same family within a genus or across taxa?
So in the papers we have some simulations that we exactly looked at what is the amount of sequence divergence that is needed in order to get the best results. So, but again, you get using simulations we get the obvious result that if you look at species that are really close together, then you don't have enough information because you don't really see. So in this case, you will need many more species in your analysis. And the same if you look for sequences that are very widely apart, the power is lower. But I think you can play with the method and find out. In the back.
To what extent you can extend your method for quantitative traits?
Currently, it's only for discrete. There is one method by Pujol and Latilot that look for continuous traits. MB 2011. I think it will be a nice extension.
Thank you again.
Thank you.
genes is way higher than what we see with the long non-coding RNA, with way darker colors here. And we also see that, as we saw with the principal component analysis, the different tissues here cluster together. But when we look at very similar expression pattern for the protein coding genes, they still are very distinguished one from another, but it's not the case with the long non-coding RNA, for example, here. Then we also performed the principal component analysis per tissue this time, and we see a very strong phylogenetic signal in our data. So we really see that the different sample cluster according to the tribe they belong to. We also see that for some tissue, there is a difference in expression between the two different sexes, so male up here and female down here, but it's not the case for all the different tissues. So here, for example, for the LPJ samples. And if you would like to know more about sex-biased gene expression, find Nico tonight at the poster session. He can show you a bit more. And then we also wanted to see how is gene expression evolving through time. So we decided to plot the correlation values that we had between all the different pairs in function of the genetic distance existing between the different species in the pair. And we can see a general trend that was expected, namely that the species that are closely related have a very similar expression pattern, while the species that are most distant have also a less similar expression pattern. So the majority of the pair follow this general trend, but we also see some interesting case where the pair do not follow this general trend. So this, what I showed you, was just for one tissue, for the ovary. So now if again we separate the protein coding from the long non-coding RNA and we present the different tissue, we see that again, as we saw with the heat map, that the expression correlation is higher for the protein coding genes compared to the long non-coding RNA, but also that there is a difference between the tissue, where we have here brain, LPJ, and the gills higher than the liver, the testes, and the ovaries. Then we also calculated the rate of expression changes with expression phylogenies. We did that for all the different tissues separately. The topology of the expression phylogenies that we obtain are very similar between the tissue, but we see variation between the branch length obtained for the different expression phylogenies. And here, if we represent the median branch length for the different tissue and separating again protein coding from long non-coding RNA, we see again that the branch lengths are higher in the long non-coding RNAs compared to the protein coding genes. And again, that the brain, the LPJ, and the gills cluster together, and that the liver, the ovary, and the testes, again, with higher branch length. We also wanted to know how many genes are specifically expressed in one tissue and if there is difference between the different cichlid striae that we have in Lake Tanganyika. So here you have the number of tissue-specific expressions, so the amount of genes that are exclusively expressed in one tissue, and we see that the testis is the tissue with the more gene expressed, with the more genes specifically expressed in his tissue, and then the brain. We also see some difference between the tribes, but there is not one specific tribes that pop up in all the different tissues. So with this, I would like to conclude that we saw that there is a high level of conservation among the organs. We could see with the principal component analysis, but also among the tribes. We see difference in selection pressure between the coding and the non-coding part of the transcriptome. We always have a higher correlation for the protein coding compared to the non-coding part of the transcriptome, but we also see some difference among the organs. And with this, I would like to thank everybody that helped me during that big project, especially Walter, Astrid, and Anouk, and you for your attention. Thank you.
Any questions from?
Yes.
I don't think this is working. Yeah, there we go. So, do you think there's some biological system in these fishes that is really responsible for their adaptive radiation? Like, is there any specific biological system, like, I don't know, does the immune system help them to spread to different environments and then radiate adaptively like that. Do you think there's any specific system that's really responsible?
Yeah, I think there is something special about it. We don't exactly know what it is for sure. And with my project, we're trying to figure out if it could be related to gene expression. And I see that what I was showing, that not all the different tissue behave the same. So this is why. But I think it's also very different about the question. Like, for example, we know that the lower pharyngeal jaw is very important for food, for example. So there we would expect something else than, for example, in the gills that could be related to immunity, for example.
Nice talk, Ahmed. I noticed in the sort of phylogeny of the expression, there was one in the testes that had really a long branch length.
Yes.
Is there anything interesting about that lineage in terms of sexual selection or competition?
So this, in parallel, I'm working with Astrid on identifying new sex chromosome, and this is a species where we have a very strong sex chromosome. So I think something is also happening there because of this. Yes?
Thank you for the talk. And have you ever checked gene ontology enrichment tests?
Yes, I'm doing that a lot. But the thing is that we don't have gene ontology for the Nile tilapia that I'm using. So I'm using other gene ontology related. I'm blasting the genes and then I use. But of course, it's very biased to what is annotated. So until now, I didn't find anything that could really be useful for my project. But I'm trying, yes.
Any other question?
Hey, have you thought about trying to do these analyses where you try and link variation in gene expression within a tissue to the ecological variation across
Yes.
species?
So I'm doing this, and I was thinking I'm gonna have time to present that, but it was too short. But yes, because we have information about, we have morpho information for the lower pharyngeal jaw, for example. So I'm really trying to correlate the morpho data with the expression data to identify some genes that could be related to the phenotype. And I'm doing this also we have information about depth, so I'm doing that for the gills, for example. We have information, we have isotopes. I'm trying to look if there is something maybe with the expression of the liver. So I'm doing that in parallel, yes. And we have some candidate, but I mean, the point will not be to, I mean, it would be nice to test them, but we are not there yet, so.
Any other one?
Yes.
Thanks, really great talk.
Thank you.
For the testes, do you have any ideas why the gene expression is so high for the testes with respect to the other organs?
Why it's so specific to that?
Specific, yeah.
Yes, we have some idea. I can maybe talk with you later.
Thank you.
Thank you. Okay. Hello. Our next speaker is Denise. She's going to talk about differential gene expression in CAST and sex-specific gonad development in honeybee.
Okay, hello, good morning everyone. I'm Denise, and today I will present to you part of my PhD work that is the differential gene expression underlying CAST and sex-specific gonad development in the honeybee Apis mellifera. So, as you may know, in these colonies from these bees, we have the females that develop from diploid eggs and the males developing from haploid eggs. But even developing from the same kind of egg, we have the workers that are facultatively sterile and does not mate, while queens are highly fertile and mates with 10 to 40 males. And the males are also called drones, and they are also highly fertile, but they have the only function of mating with one virgin queen. So when we think about this mating strategy, we think about the gonad phenotype. And in the genus Apis, we have what we call exaggerated gonad size. So we have workers that have 2 to 20 ovarillus per ovary, and this is in result of programmed cell death during development, and queens with 150 to 200 ovarillus per ovary, laying up to 2,000 eggs per day. Males are really similar to queens, having 150 to 200 testes per test. So, really similar. When we think about Apis against all the other social species of bees, we see that all the sex and caste have only 4 to 8 ovarillus, or testes per gonad. Other interesting thing that is different between them is that Apis are extremely polyandric, with high number of matings, against all the other social bees that are essentially monandric, with single mating. So this leads me to my main question. What are the developmental molecular mechanisms underlying the evolution of this exaggerated gonad size that we see in the genus Apis? So for this presentation, I have 3 main hypotheses that are distinct from each other. The first one, I expect that Apis mellifera drones and queens have similar gene expression pattern related to gonad development. As we see, they are really similar morphologically. The second one is that genes related to gonad size may differ between Apis and Bombus. Bombus terrestris, in this case, is a primitively social bee, and they have only 4 ovarillus per ovary, or testes. So the third one is that those genes related to gonad size evolved under diversifying selection in the genus Apis. So this may be the reason for this exaggerated gonad size. So to test this hypothesis, we decided to run RNA sequencing in gonads from Apis mellifera and Bombus terrestris larvae. So we collected ovaries from workers, ovaries from queens, and tests from drones, 3 samples for each. And we decided for a critical stage in divergencing queen ovary size, queen worker ovary size. And for Bombus terrestris, we collected 3 samples from gonads in the respective stage. And after RNA extraction from the samples, we sent it for sequencing in Tübingen, Germany. So the first results are regarding the principal components analysis in the gonad, Apis mellifera gonad transcriptomes. So here we see in green, the worker samples, in red the queen samples, and blue the 3 samples from drone. Here we have 2 overlapping. So they cluster together, but when we look, queens and workers are more similar to each other. And this goes against my first hypothesis that we were expecting that queens were more similar to drones. Okay, and then we decided to look for the different expressed genes. And here I have a total of 8,255 different expressed genes. And here we plotted in this Venn diagram our 3 contrasts of comparisons. And as we saw before, the ones with drones, the contrast with drones, are the ones with higher number of different expressed genes. Here in the center, we have 870 genes. These are genes that are different expressed in all the contrasts together. And as we wanted a profile for Apis mellifera to compare with Bombus terrestris transcriptomes, we decided to start with these 870 genes. So we run with them a gene ontology enrichment analysis, and here we plotted only the most significant categories. And we had a total of 7 enriched categories related to oxidoreductase activity and binding activity functions. And the plot here, the size of it is related to the count of genes inside of the category and the color regarding the significance of the enrichment. These 7 categories represent a total of 48 genes. And with those, we were finally able to run a comparison with the transcriptomes from Bombus terrestris. So we run a clustering analysis with those 48 genes against their Bombus orthologs. And here, first, I want to show you the clustering of the samples. So I have the 3 samples from workers, 3 from queens and drones, from Apis mellifera and Bombus. So Apis clustered together, Bombus also different from each other. But when we have a look to the difference inside Apis, for those 48 genes, we have that drones and queens are more similar to each other. So for those genes, maybe they are related to the gonad size in Apis mellifera. So as we wanted to see, and we have the third hypothesis, that Apis evolved different in genes related to gonad size, we looked for genes that were different between Bombus and Apis. So we finished with 3 clusters of genes that are different between Bombus and Apis, and they represent 17 genes. And then with those, we looked for the coding DNA sequences in 13 species, these ones, that we have published genomes. And after aligning these 13 sequences for each gene, we were finally able to run an evolution and look for evolutionary signatures. And we used Busted, that is implemented in DataMonkey. And what is interesting about this test is that you can select the branches that you want to test. So in case our hypothesis against Apis, so we selected as our foreground branches all the 4 species for Apis, and against our background, all the other 9 species. And the result is this one. So we have one gene, the first one, that has DNDS higher than 1. So it's really positive and when we think about diversifying selection. But we also have positive result for other 3 genes. And here I come again to the test. As we selected the branch sites for Apis, we have a significant evidence that those genes have evolved under diversifying selection in the genus Apis, and this is significant when we compare to the sites that are under diversifying selection in the background. So here I just show you the location of those genes in the clustering analysis that we had before. So they really differ between Apis and Bombus. And unfortunately, I didn't have time to go deeper inside of those genes. Sorry. Okay. And the conclusions for this talk is that the transcriptome in Apis mellifera is more similar between females, not between drones and queens, as we expected. However, the expression of those genes with significant gene ontology categories is more similar between drones and queens. And this maybe is evidence of the relation to the gonad size in Apis mellifera. So at least 4 genes from the genomic comparison Apis versus background showed significant evidence of diversifying selection. So with those 4 genes, we want to go deep and also verify their expression during development in Apis mellifera. And of course, we still have a huge transcriptome, and this is quite new data, and we want to check all the other different expressed genes contrasts in Apis mellifera transcriptomes, and also look for evidence of selection. I want to thank my professor Klaus Hartfelder, that is my advisor since my master, is here. And also my supervisor in Germany, Professor Dr. Martin Hasselmann, and his group that received me in Germany for 1 year. And thank you for your attention.
Thank you, Denise. There are any questions in the audience?
Thank you for the really interesting talk.
Thank you.
I was wondering if you had expression data from other tissues in the bees, and if you could contrast the other tissues, the differences between the drones and the queens and the workers. Maybe some of the differences that you're finding in the gonads are global differences, and then maybe you get. I was wondering if you had those tissue.
Yeah, right now, no, because our focus was looking for evidence of selection in gonads, but we also have other bees transcriptomes, so we are first focusing gonad transcriptomes, and then we will look for other tissues.
Okay, thank you.
Yeah, so following this idea of selection, I wonder how you conclude that these genes were selected for, because, well, you showed the cytochrome P, which were implicated in the coloration, actually. So I was wondering if there were any, like, you know, morphological differences between the gonads, maybe the color or whatever, or could it be also connected with, you know, overall color of the bees or so? I don't know. Doing transcriptomics analysis, it's sometimes a little bit difficult to prove that it was indeed selected, not like, you know, randomly just differentially expressed because of any reason. So if you have any comment of that, we'll appreciate it.
Yeah, it's quite difficult to say that they are really having a function on here, because they are actually really general genes that work there, and we are going deep on them now to see if they are really related to gonad size, but maybe in this phase they have some functionality in the gonad size. We don't know yet. Go back there.
It might have been also interesting to try to do on these 4 candidate genes, like, you know, qPCRs in different tissues, and then you could see if you have differential, if they are differentially expressed in only gonads. That could be interesting.
Yeah, now that we are thinking about the gonad development and going deep in the gonad development, and also we have the samples already for in other tissues, so must be a nice idea to run this in other tissues. Thank you. Another question.
Any other question?
Is there much variation in gonad size within the genus Apis to add more sort of phylogenetic contrasts?
So I didn't test yet within genus Apis, but yeah, probably because Apis florea, for example, they have a smaller number of matings and also a smaller gonad size. So maybe there is a difference there.
Thank you very much.
Thank you. I'm taking off my glasses. Yeah, it's
Wait, you also have the thing in your pocket.
I'm bringing it.
No, I haven't. Okay, so you just put it in your pocket. Like anything you want to know before the presentation starts?
No, I don't think so.
Always good.
Thanks. I hope it's not gonna fall. Spero che non cada. Spero avere gli occhiali. Ok.
Hello, everyone. Our next speaker is Chiara. She's going to talk about evolutionary perspective in domestic avian species.
Thanks for the introduction. So today I'm going to share with you some of the research I've been doing during my PhD. And it involves food feathering in domestic avian species. So parallel evolution is the independent development of the same trait in two different species that share the same common ancestor. And the trait is not observed in a common ancestor. It might be very simple, and it might involve similar wing spot patterns in, for example, in divergent lineages of butterflies. But it can also be much more complex, like, for example, similar morphological structure of the camera eye in cephalopods and vertebrates. Parallel evolution is usually quite common in vertebrate evolution. However, the lack of an appropriate model species to study convergence from a genomic perspective has challenged our understanding of parallel evolution. And the first step forwards was actually achieved in 2004 when the chicken genome was published. And this, of course, represented a very important milestone because this was the first non-mammalian amnio species to have the genome sequenced. And of course, researchers had a lot of hope to use this genome to actually get a better understanding of vertebrate genome evolution and also to get a better annotation of the human genome. So chicken is actually a very important model species, not only to study vertebrate evolution, but also to study the complexity of traits. And today, I'm going to share with you the genetic basis of a very interesting trait, which is called ptilopody, or foot feathering. And this trait involves the development of feathers at the level of the ankle and foot, and can be observed in both domestic species, but also in wild avian species, like snowy owl and ptarmigan. And this trait is usually not observed in the ancestor, which you can see on the top left. Actually, the genetic basis of this trait has been under discussion for many years. However, the genes and chromosomes that are directly involved in this phenotype are still unknown. A better understanding comes actually from another domesticated species that show exactly the same phenotype, and this species is the pigeon. And in a recent study in 2016, Domyan et al. Associated this phenotype to two genes, PTX1 and TBX5. So if we take pigeon also as a reference, of course there are some questions that arise. And the first one is actually this phenotype caused by the same genes also in chicken? And if it's really the case, are we also talking about the same underlying mutation? And if it's also the case, are also the same regulatory mechanisms behind this phenotype? So to answer these questions, we use whole genome sequence data from 169 individuals. And we performed a genome-wide association study. And as you can see, we actually found two main genomic regions associated with the phenotype, one on chromosome 13 and one on chromosome 15. When looking into more details on chromosome 13, we found protein coding genes H2AFY, which was also found previously in pigeon. And we also identified a long non-coding RNA and a QTL that was found by Sun et al. As linked to the phenotype. We also looked at structural variants in this region, and we indeed observed a 17 kb deletion upstream of our candidate gene. And interestingly, not all foot feather samples were actually homozygous for the deletion. We then look into more detail at the deletion, especially the breakpoints, and we actually observed a 7 base pair microhomology, which shows different level of conservations across different bird species. On chromosome 15, we actually identify one protein coding genes, TBX5, which, as you remember, it was found also in pigeon to be associated with a phenotype, and a long non-coding RNA, and once again, another QTL that was found by Sun et al. In 2015. We performed a structural variant analysis also on chromosome 15, but we did not find any significant structural variant likely associated, so we performed a haplotype analysis. And the haplotype analysis, as you can see, clearly distinguished the foot feather birds from the ones that did not have the phenotype, which suggests that foot feathering has evolved only once in chicken. We then looked into more detail at the evolutionary history of this trait by using the multiple sequence alignment generated by Green et al. In 2014, which included 15 birds, 3 crocodiles, 4 turtles, and an Ollie lizard. And specifically, we look at conserved elements on chromosome 13 and 15, and we found that some of our variants were actually found in conserved elements. So a little bit of discussion now. So even though chicken and pigeon diverged more than 69 million years ago, it is really amazing to see that the same phenotype is likely determined by the same genes and also underlying genetic basis. And this trait has been actually extensively selected in both species over time. And it also associated with very different variation in terms of type of feathers and length. And this trait has also become nowadays a breed characteristic that breeders are actually actively selecting for. When looking at the deletion, so we found a 17 kb deletion, and our deletion was actually completely overlapping the 55 kb deletion that was found in pigeon. So we also have here the same mechanisms. And interestingly, as also was shown in pigeon, not all birds were actually homozygous for the deletion. And even though the PITX1 was not found within our peak, is actually found very closely upstream of our candidate gene, H2AFY. And what we think is going on is that the deletion is, yes, important for the phenotype, but is actually acting like a modulator to actually modify the type and extent of feathers. Regarding the haplotype analysis, so we show that actually the haplotype origin is exactly the same, and it has evolved only once in the domestication history of chicken. And interestingly, our haplotype was actually found in the London coding RNA, which is only 6 kb upstream of TBX5, which is the candidate gene. However, we cannot actually say that this London coding RNA is directly involved in determining the phenotype until we do not have RNA-seq data. And indeed, we are actually working now on verifying this hypothesis using RNA-seq data for, of course, validation. So to conclude, we have shown that pilopody is determined by the same genes and mechanisms in pigeon and chicken, so it's a clear indication of parallel evolution. And the trait has evolved only once, and as already mentioned, it has evolved through parallel evolution in the 2 species. So with this, I would like to thank my collaborators, Wageningen University and the Image 2020 Horizon for the funding. And I will be happy to take any questions, if any.
Question in audience?
Yeah.
Hi, thank you. On just the phenotypic level, do the scales turn into feathers, or are the feathers added? Do you keep the cell type that makes the...
What do you mean exactly?
Like the ancestral type has like scaly
scale
structures?
yeah.
Yeah, do they turn into feathers, or is it that the feathers are added?
The feathers, we think that the genes for the ones that have scale epidermis is actually not active.
Okay.
So it's actually never present. Although for the ones that have the foot feathering, you can clearly see already at an early embryonic stage that the feathers are growing on the feet.
So you did developmental series for the...
Yeah.
Okay, cool.
Yeah.
I actually have 2 questions. One is shared with the colleague here on my side. The first one is, do you know if the 2 things interact in some way? Like, are they part of the same developmental network or whatever? And the second one is, why are they selecting for more feathers in the feet? I don't see any advantage for the producers to have more feathers. It's like annoying, isn't it? Like, why?
Okay, so I will start from the last one. So they actually select for this trait just because it's fancy and it's beautiful to see. So it's mostly for showing purposes, because these are all traditional breeds that they are just kept for fancy and hobby breeding. So of course, people just like it and then they start selecting for it. And the interesting thing is that this trait is actually quite easy to select for. And then for the first question, so you mentioned the 2 mechanisms. You mean the one on chromosome 13 and 15? We think so, although we will actually like to see the RNA expression, how it actually changes because we have sampled now 2 different embryonic stages of development to see whether these 2 genes, for example, are up or down regulated also in different stages of development to see actually what is the interaction between the 2. Yeah.
So following up the RNA-seq, are you planning to also do some functional experiments to prove exactly how the mechanism works?
Yeah. So I already performed a functional analysis on our on the significant variants that we found on both chromosomes, and they're actually all, interestingly, and I'm happy to say that, non-coding. And so the only thing that you can get is intronic variant, upstream gene variant, so you actually do not get the function directly of that. But we actually developed methods that can score the non-protein coding variants based on their potential impact on the phenotype and the individual. And actually, the score is quite high for most of the variants, which means that they actually have a function and they impact the phenotype. But yeah, the paper is still not out, so it's in preparation.
So I was just curious about the prevalence of the foot feathering across the bird lineage as a whole, because the 2 species you mentioned are pigeons and chickens, which are heavily domesticated. But
yeah.
You mentioned the snowy owl as well.
Yeah.
So this is a trait that obviously occurs naturally. So if there's a potential of taking a broader phylogenetic perspective, would that be possible? I don't know what the distribution's like.
So for in the wild, some of the raptor species actually have this phenotype. So not only snowy owl, but also the ptarmigan, and it's mostly for thermoregulation. If you think, for example, of the snowy owl, so there's a logic function for that. So that is clearly under natural selection. This is mostly on artificial selection. But it's also very interesting to see that even though you have 2 different type of selection, the trait is exactly the same. And what I think is that the mechanisms that we observe in chicken and pigeon most likely are also responsible for the phenotype in the wild. So it's actually something you can trick easily, actually. Yeah.
Wait for the
Sorry?
Is there any evidence that the trait is polymorphic in the ancestor in red junglefowl?
So what we think, based also on recent discoveries of fossil records, is that what you usually think is that the red junglefowl, so the wild ancestor, is actually so the scaleless is actually the ancestral one, and in this case the foot feathering is the derived trait. But based on fossil records, it seems that it's exactly the opposite. So this is the ancestral, and the wild is the derived form.
Okay, thank you, Chiara.